afternoon, everyone. So we're going to get started with our afternoon session. And it's a real pleasure for me to introduce the first speaker of our afternoon session, and that's Dr. Cristina Nostro. Cristina is a scientist at the Toronto General Hospital Research Institute. She's been here since 2012. She's also a scientist within the McEwen Center of Regenerative Medicine and holds the Harry Rosen Chair in Diabetes and Regenerative Medicine Research. Her work in the laboratory is focused on the generation of beta cells from human embryonic and induced pluripotent stem cells, which is what I think she's going to tell us about, where she's made some very important advancements. Cristina, welcome.
Thank you, Mansour, and I would like to thank the organizers for the opportunity to present. This is work that was recently published, but I'm going to end with some unpublished data as well on, how our approach is on how to understand how pancreatic development happens and use this information to develop therapies for diabetes. So as you all know, diabetes is a global emergency. About 350 million people are affected worldwide, and this number is increasing. About 10% of the population is affected by type 1, and the current treatment for type 1 is, I don't know whether it's, yes, you see the mouse. The treatment is, of course, insulin injection with either needles or pumps, or transplantation, or all pancreas transplantation or islet transplantation. The problem with this is, of course, donor scarcity, as well as the variability of the tissue, whether it's the pancreas or the islets. Every donor will be different, and in particular for the islets, most of the time you will need 2 to 3 donors per patient. So the idea is to generate pancreatic tissue, whether it's like progenitors or islet-like cells, from human pluripotent stem cells. These are ES or iPS cells, and transplant them into patients. This is already happening. There are clinical trials on the go. And as you all know, human ES cells have the potential to generate all the lineages in the body. And we are interested, of course, in generating the pancreas. And I don't want to go into the details, but essentially this is work that spans approximately 12 years. It's not just our lab, but it's many labs around the world. We have been able to recapitulate what happens during embryonic development and take human embryonic stem cells as well as induced pluripotent stem cells and differentiate them through several steps into what we call a pancreatic progenitor cell. This is a cell that has the capacity to give rise to beta-like cells in vitro as well as in vivo. The same cells that expresses PDX, if you want, this pancreatic endoderm, can be converted in what we call a polyhormonal cell. This is recapitulating early stages of development, and these polyhormonal cells will give rise to alpha-like cells. I want to take a moment to describe these two populations because it's going to be important for the talk. These two cells are mainly different by the expression of a marker that is called NKX6.1. As you can see here by flow cytometry, both the pancreatic progenitor that give rise to beta cells as well as the polyhormonal cells express PDX1 at high level in a high percentage, but only the pancreatic progenitor cells has cells that co-express both PDX and NKX6.1, but while the polyhormonal do not have this double positive population. And also, as I said, the pancreatic progenitors do not express insulin at this early stage, while the polyhormonal cells do. Just to put it into context, these pancreatic progenitors, when transplanted into an immunocompromised mouse, will give rise to all the lineages of the pancreas. What you see here is graft coming from this, from the human ES cells progenitor, human embryonic stem cell derived progenitors transplanted into these mice, and they will form 5 months after transplantation beautiful islet-like structure. You see here insulin in green, glucagon in red, somatostatin, and pancreatic polypeptide. So all the lineage, all the hormone positive cells that you will find in an adult islet, in an adult pancreas. at the same time, this pancreatic progenitor will give rise to cytokeratin 19 positive cells, which would be the equivalent of ductal cells, as well as trypsin positive cells, which are acinar cells. So in a way, what I'm showing you here is that the pancreatic progenitors that we generate in vitro has the capacity to give rise to all the lineages of the adult pancreas in vivo. I don't know at this point whether it's one cell that has the multipotent potential or whether it's like several or individual unilineage progenitors that give rise to all the different lineages. Just again, to put this into context and bring you up to speed in what's happening in the field, similar cells, pancreatic progenitors, are now being used in the second clinical trial launched by a company called ViaCyte, and where the ViaCyte is transplanting these pancreatic progenitors inside the device. And this device now is perforated so the vasculature can go inside and nourish the pancreatic progenitors. This is very important because in the very first iteration of their clinical trial, the device was not permeable to the vasculature, and in that particular case, there was foreign body response, and the pancreatic progenitor cells inside the device did not mature. This means that now, of course, in this particular trial, they are using immunosuppressant, and the potential of this is that cells within the device can potentially escape if there is a tumor. And this is where our work comes in, because we can be very efficient at generating pancreatic progenitor. What you see here is that production of over 90% positive NKX6.1 positive cells, which are progenitors. However, this is not always the case. When we analyze several different human embryonic stem cell line and iPS line, we see a variation in the generation of these progenitors. So sometimes we can be as efficient as 90%, and in other cases, as you can see here in this iPS line, we can only generate 40% of pancreatic progenitors. So the question is, if we want to transplant these cells into patients, we need to make sure that we have a population that is safe. And there are two ways to do this. One, we can optimize the differentiation and make sure that we always get the right number, or we can try and figure out a way to select the pancreatic progenitor population and purify it. So this was the question that we had. Can we actually generate a safe cell population that one day could be used in transplantation without a physical barrier separating the cells from the host? And the hypothesis we had is that if we compare the pancreatic progenitors to other lineages, we should be able to identify cell surface markers that can allow us to purify this population using either fluorescence-activated cell sorting or magnetic activated cell sorting. So to do this, we actually started a collaboration with Thomas Kisslinger at the Princess Margaret Cancer Center. He's a mass spectrometry expert. And we provide Thomas with three populations: the undifferentiated human ES cells, the pancreatic progenitors, and the polyhormonal cells. And the rationale for this was because these cells are very similar, especially the pancreatic progenitors and the polyhormonal cells are very similar, so the difference that we would detect would be really specific. And with Thomas and Ankit, the student in his lab, we, they digested the proteins into smaller peptides, and we focused on glycosylated proteins. The rationale for this is that every protein that is glycosylated is destined to be found on the membrane, whether it's the cellular membrane or internal membranes. This way, we could focus on cells that could be specifically found on the surface of the cells. So the glycosylated proteins were enriched with magnetic beads and bound to a column. The unbound fraction was washed away, and we removed the magnetic bead from this glycosylated peptide by deglycosylating them and ran them through the mass spec. The result for this was very satisfying because, as you can see here, the hierarchical clustering analysis showed that we ran the samples in triplicate, so we have the triplicate undifferentiated human ES cells on the left, clustering far away from the pancreatic progenitor cells. And the pancreatic progenitors are separated from the polyhormonal population. More importantly, if you look at this, there is a cluster analysis that identified peptides that are specifically expressed in the undifferentiated ES cells here. This is cluster 1, where you see depicted here, as well as clusters that were specifically enriched for pancreatic progenitor cells or polyhormonal cells that you see here, and so forth. So, of course, we focused on the peptides enriched in the pancreatic progenitor cells. And among this, we mined the data looking for proteins where we could find antibodies for flow cytometry that were commercially available. One of these was GP2. As you can see here, we validated the mass spectrometry data using flow cytometry, and what we saw by mass spectrometry was validated perfectly. As you can see, GP2 is not expressed in the undifferentiated ES cells, is expressed highly in the pancreatic progenitor population and not found in the polyhormonal cells. What is GP2? GP2 is the pancreatic secretory granule membrane major glycoprotein 2, and it's a protein that is found on acinar cells in the adult pancreas. This is a picture coming from Human Protein Atlas. As you can see, the brown identified GP2 staining in all the acinar tissue. If you're familiar with immunohistochemistry from pancreas, you realize that these are islets and they are negative for GP2. This was, in a way, a little bit puzzling in the beginning because we expected this marker to also mark maybe the beta cells. So in vitro, we find it associated with the pancreatic progenitor, cells that will give rise to all the lineages of the pancreas. But in the adult, it's only found in the acinar tissue. So we ask, is this pancreatic progenitor, is GP2 really marking the pancreatic progenitors in humans, or is it an artifact of in vitro differentiation? So to answer this question, which is GP2 expressed in pancreatic progenitors during human development, we started a collaboration with a group in Vanderbilt University run by Al Power and Marcela Brisova. And what we asked them was to stain neonatal pancreas for GP2 and NKX6.1, as well as insulin. The reason for that is that we wanted to identify the cells that could potentially co-express GP2 and NKX6.1. This would be the equivalent of what we see in vitro. The NKX6.1 positive, I remind you, is the pancreatic progenitor cells that we generate in the culture. And I don't know whether it's clear, but you can see that in the middle of this neonatal pancreas, you have beautiful insulin-positive cells marked by the blue staining, co-staining NKX6.1, and negative for GP2, which is exactly what we see in the adult pancreas. So in the adult pancreas, the islet is negative for GP2. And then surrounding this, you have GP2 positive cells, and some of these GP2 positive cells, like you see here, are double positive for NKX6.1, suggesting that indeed in the human pancreas there is a cell that is NKX6.1 positive, so it's a progenitor that co-expresses GP2. To make sure that really these cells was what we call in the field the multipotent pancreatic progenitor, a cell in the human pancreas that will give rise to all the lineages of the pancreas, so the endocrine, the endocrine, the acinar, and the ductal, we co-stained for another marker called PTF1A. In the mouse, it has been shown that PTF1A and NKX6.1 co-staining marks these multipotent progenitors, which is normally found in the tip of the pancreas, which would be this area here. This is the equivalent of the trunk. And as you can see, PTF1A positive, NKX6.1 positive, and GP2 positive cells are found in this structure, suggesting that really what we see in vitro is recapitulating the event of leading to pancreatic development in vivo. And this was actually the first time these cells was identified in humans. So we then went back to the culture to identify when does GP2 come up in the differentiation. And as you can see here, by day 10 of differentiation of human embryonic stem cells, we start to see GP2 just before NKX6.1 becomes expressed. And the cells making expressing NKX6.1 and double positive for GP2. Usually by day 13 of differentiation, we reach our peak of NKX6.1, and these are all positive for GP2. This data that I just showed you was generated using a reporter cell line where GFP was under the NKX6.1 promoter. We also made sure that this was true in an untargeted line where we didn't have GFP under NKX6.1, so we co-stain with antibodies against NKX6.1 and GP2. And as you can see here, we have double positive cells, which also co-stain PDX1, which is another marker of the pancreatic development. So this was all great, but the question that we had was then, can we actually use the GP2 positive cells to generate pancreatic, to generate beta-like cells in vitro? Because if you purify cells from differentiating culture, maybe then you remove cells that would cross-talk to the GP2 positive cells and be important for differentiation. So it was important to demonstrate that GP2 positive alone could make beta cells. So we did just that. We sorted for GP2 using fluorescent activated cell sorting, and then we repeated the same experiment using MAX. And we could sort for the GP2 positive cells and culture them in culture conditions that were recently identified by two groups to induce beta-like cell differentiation in vitro. And indeed, we could show that using GP2 positive cells, we could generate a higher number of NKX6.1 positive C-peptide positive cells, this double positive population here, which marks the beta-like cell. So a beta-like cell in vitro is characterized by the expression of these two markers, C-peptide as a surrogate for insulin staining and NKX6.1. As you can see, we generate higher percentage of these beta-like cells from the GP2 positive fraction compared to pre-sort and negative fraction. And these GP2 positive cells will give rise to cluster of cells that contain C-peptide positive cells that have very few glucagon positive cell contaminants, no acinar tissue, and pretty much equivalent to the pre-sort population. And when we tested for these cells for their functional property, the usual test is a glucose stimulated insulin secretion assay. And I'm not going to show you the data because we couldn't really generate functional cells in vitro. So these beta cells are still immature. And the reason for that is that we think that these cells actually lack the glucose transporter, GLUT1, which is the transporter that is found in human beta cells. The mouse beta cells would have GLUT2. And I want to show you something here, which is kind of really new in the field. I think we're the first one to really look at this level. You can see there in the pre-sort we have insulin positive cells as well as GLUT1 positive cells, but the insulin positive cells do not express this glucose transporter and therefore cannot sense glucose challenges and release insulin. And I think we need to generate a cell that co-expresses both insulin and GLUT1 to be able to have a functional cell in vitro. So we're working heavily on this. In the same way, you can see that the GP2 positive cells generate insulin positive cells also not expressing GLUT1. There is one GLUT1 positive cells, but it's not co-expressing insulin. So I think these are the effort in moving forward. But the next question that we have, which is leading me to the unpublished data, is can GP2 cells give rise to beta cell in vivo? This is an ongoing experiment, and I'm just going to show you some preliminary images. We sorted for the GP2 positive cells, and we transplanted GP2 positive cells and unsorted cells into immunocompromised mice. And occasionally, in the unsorted fraction, we detect big outgrowth. This is how the mice look. This is the same mouse that you see here. There is an outgrowth in the mice transplanted with the unsorted cells. We never see an outgrowth coming from the GP2 positive cells. These mice are still alive. The ones transplanted with the pre-sorted cells were sacrificed, and indeed we found tumors. And this is consistent with what has been published in the field. There is an average between 15 to 40% of the pancreatic progenitors derived from human embryonic stem cells will give rise to tumor. we are now suggesting that sorting for GP2 will give rise to a safe population that will eliminate the tumor formation. And we have also data showing that indeed in the GP2 positive cells, we have cells that seem to be responding to glucose challenge. And with this is, I'm going to the conclusion. I'm showing you that glycocaptured proteomics identified GP2 as a novel cell surface marker that tracks the emergence of these pancreatic progenitor cells in vitro. And we believe that these cells is the same cells that we found in vivo in neonatal human pancreas. We've shown that GP2 sorted cells can give rise to beta-like cells in vitro, and we now have data suggesting that by sorting for GP2, we can purify the population and give rise to a safer, probably, population for treatment of type 1 diabetes in humans. And I would like to thank, obviously, the people behind the work. Some past members, Catherine Cogger started this project, but we had a lot of help, especially from the lab of Thomas Kieslinger at the Princess Margaret Cancer Center, the group in Vanderbilt with Marcela Brisova and Al Power, and a group in Portland, Oregon. And with this, I'd like to thank you and take questions. Mina.
Beautiful work, Cristina. Mike's coming. So as always, beautiful work. So as you know, there are ways to transdifferentiate from the ACE&R back to beta cell-like. And I might have missed this, but would that have anything to do with the GP2, that the fact that GP2 is expressed in the exocrine cells, and could that play a role in that context, perhaps?
We haven't explored this, but I think it's something that it would be really interesting to look at in specific models where you see transdifferentiation happening, or in even in type 2 diabetes. What happens to the GP2 population? Do we generate more GP2 positive? There are, that have the capacity to give rise to beta cells or transdifferentiate, or even like there is all this theory about, in type 2 diabetes, there is dedifferentiation of beta cells. Would they revert to a GP2 positive cells? We haven't looked, but I think I have a feeling that Al Power in Vanderbilt is looking at samples of type 2 diabetes to patients to see whether he sees differences in the GP2 positive population. But it's an interesting point. What happens not in normal development, but in the pathophysiology? Would the GP2 population change? Yes.
Hi. You mentioned that your GP2 positive didn't express GLUT1 and yet were responsive to glucose. So did you look for other glucose transporters?
So no, the insulin positive cells will lose the expression of GP2, but in our hands these cells don't have GLUT1. But to answer your question, no, we haven't looked at other glucose transporter, and we should, because maybe this dogma that only GLUT1 is found in beta cells is not real. So I really need to look at that. It's a good point.
Cristina, you mentioned in your strategy that you selected from the profile those proteins against which antibodies were commercially available. Were there any other interesting targets that you sort of need to develop an antibody for because they actually might be more revealing?
Yes, and in fact, which brings me to your talk earlier, we actually submitted an IP application with the Tech Transfer Office to cover for all the other markers that we identified. In particular, I think it would be interesting to look at the markers that are not expressed in that population, potentially to go into lineage depletion.
And coming back a little bit to what you were talking with Mina about, both in the acute phase of the onset of type 1 diabetes and maybe even in type 2 diabetes, sort of the ongoing loss of beta cells, any way that some of your findings can be biomarkers for cell turnover or quantification of pancreatic cell mass, which has eluded, for many years, diabetes research, right? To be able to quantify someone's beta cell reserve. Any thoughts about that?
It's interesting, actually. I was talking to somebody about looking into serum from a young patient with type 1 to see whether GP2 would be detected earlier on as a biomarker, as you say, to maybe detect early onset. It would be nice.
Yeah. Any other questions for Cristina? All right, well, thank you very much. Beautiful talk. It's my pleasure now to introduce someone who needs no introduction, our division head in advanced diagnostic and senior scientist, Myron Cybulsky, who also is a Canada Research Chairholder and the winner recently of a foundation grant from TGHRI. Myron's been working for many decades in atherosclerosis and continues to make important contributions in this area. Myron's going to share with us his most recent findings.
Thank you, Mansour. Let me just get set up here. So today I'm going to talk or tell you about some of the research that we've done over the last several years and some of the directions where we're heading. So my talk will be divided into 4 components. The first is the regional distribution of atherosclerosis and the biology of the normal arterial intima at sites that are predisposed to atherosclerosis. The role of myeloid cells in the normal arterial intima at sites that are predisposed to atherosclerosis, the initiation of atherosclerosis, and some future directions. So I usually like doing research to figure out how things work. But when writing grants and trying to justify my research to fellow scientists, I try to underscore the fact why we study early stages of atherosclerosis when it's well known clinically that significant problems associated with atherosclerosis arrive in advanced atherosclerosis, usually when these human plaques get disrupted and one develops thrombotic complications that give rise to events such as myocardial infarction or embolization and stroke into the brain. So I say that understanding mechanisms underlying the onset of atherosclerosis may provide strategies that can delay or abrogate disease progression and may eventually reduce thrombotic complications. Furthermore, in clinical medicine, we often find that therapeutic interventions are often more effective when instituted at early stages of a disease. And finally, unlike in animal models where they're driven by a relentless high level of hypercholesterolemia, in humans, atherosclerosis may be much more of a relapsing, remitting disease. And some of the phenomena that we see in early stages in atherosclerosis can be recapitulated in later stages. So for example, here you can see an advanced atherosclerotic plaque with a large necrotic core and a well-developed fibrous cap. But if you look at higher magnification here at the edge of this fibrous cap, you can see that there is accumulation of foam cell macrophages within this cap. And this is a feature of early disease, early atherosclerotic disease. Right below the endothelium, you could see these accumulation of these cells. And in fact, in this same patient, there was a disruption of the fibrous cap in a non-occlusive thrombus that had formed. So I've always been interested in the fact that atherosclerosis occurs as a regional disease. Although all of the systemic factors that we're aware of that drive atherosclerosis, such as hypercholesterolemia, high blood pressure, high blood glucose, smoking, these are all systemic factors. And therefore, the entire vascular endothelium is exposed to these factors. And yet the disease develops at specific regions, as is illustrated in this en face view of the mouse aorta, showing formation of lesion in lesser curvature, which is in this animal model is the site that is one of the has the highest predisposition to atherosclerosis, and immediately adjacent to it is a site that is quite protected from atherosclerosis. And in fact, in mice, looking at normal mice, we can, in fact, knowing the endothelial biology and the cell populations that reside in the arterial intima, we can predict where atherosclerosis will develop. So the way that we study this disease process, even before it begins, is we've mapped out regions that are predisposed to atherosclerosis in the mouse aorta. We call them high and low probability regions. And then we can study these with a variety of tools. It's well known that mechanosensing and hemodynamics is an important component to atherogenesis. The vascular endothelium can sense disturbed laminar flow, which is found in regions of blood vessel curvature and at branch points, whereas straight segments of arteries experience uniform laminar flow, and this flow pattern modulates the gene expression. So way back in 2000, we studied a transcription factor called NF-kappa B, and we showed that this transcription factor was primed for activation in endothelial cells at sites that are predisposed to atherosclerosis. And what this means is simply that the transcription factor was expressed at higher level together with the inhibitor, the I-kappa B, that traps this transcription factor in the cytoplasm of cells. Much akin to sitting in a race car at a stoplight next to the little Pinto. And it's quite clear that once the light turns green, which car is going to win the race. Obviously the one with the big motor. And same here, the cells with the big level of transcription factors have a higher potential of translocating this transcription factor to the nucleus and driving pro-inflammatory gene expression. Others have shown that one of the reasons for this is that there's activation of JNK1 and ATF2, which is important for the transcription of REL-A, which is a prototypic NF-kappa B family members. And others have also shown that microRNAs are modulated in regions that are predisposed to atherosclerosis that can also modulate signaling through NF-kappa B. A number of labs have shown that there are a variety of unique signal transduction patterns, gene expression patterns, as well as epigenetic mechanisms that are located in regions predisposed to atherosclerosis. And all of these are related to the hemodynamics that is found at these sites. But in addition to differences in gene expression, we have shown that the sites that are predisposed to atherosclerosis have a different composition of cells that are constitutively located at these sites. So we found that there is low-grade inflammation at sites that are predisposed to atherosclerosis, even without risk factors for this disease, and that there is an accumulation of myeloid cells that resides in the arterial intima just below the endothelium. And by performing immunostaining, we showed that these cells express a dendritic cell marker, CD11C, and they extend dendrites and have morphological features of dendritic cells. Immediately adjacent to this is an area in the greater curvature that is exposed to the uniform laminar flow, and as you can see, these cells are not located at these sites. So our work was followed up by the work from the late Ralph Steinman's laboratory, who confirmed our findings and extended them. What they actually found was they could actually isolate these cells. They could isolate these cells from the arterial intima, and they showed that they were able to cross-present antigen to T cells in vitro, thus confirming that a functional capability of these cells as dendritic cells. Furthermore, in collaboration with Mark Henkelman's group, we could modulate the aortic, the hemodynamics in the mouse aorta by inducing aortic insufficiency through insertion of a catheter through the carotid artery and puncturing the aortic valve in mice under high-frequency ultrasound guidance. And depending on the extent of aortic insufficiency, when this was done in the LDL receptor deficient background, which is predisposed to atherosclerosis, this could dramatically alter the pattern of atherogenesis that one finds. So here on the left side, you can see the typical pattern of early lesion formation in the lesser curvature, and the more aortic insufficiency that one finds in these animals, which actually causes a flow reversal in the descending thoracic and abdominal aorta. You can now see that there's an increased amount of lesion formation in these regions. If one does this experiment in just a straight C57 black 6 mice without feeding them a high cholesterol diet and inducing hypercholesterolemia, we actually find that these same regions that develop atherosclerosis in the LDL receptor deficient background, they will now begin accumulating myeloid cells in the intima at the sites that are predisposed to atherosclerosis. And thus, we can predict where atherosclerosis will develop by looking at the gene expression pattern and the cellular composition of the intima. So what do these cells do normally in the arterial intima? So last year we published a long paper on this, showing that these cells can actually protect the intima. So the earlier study from Ralph Steinman's group implied that these cells are capable of antigen presentation, and therefore perhaps they are important in driving an immune response. Yet in order to drive an immune response, you actually need a T-cell repertoire. So in contrast to the fact that you have a relatively large number of these cells in the arterial intima, the number of T-cells that are present in the intima is very small. So there is no T-cell repertoire for antigen presentation there. So clearly this, perhaps, if anything, these cells may actually dampen immunity at these sites. So in order to, so we then created an alternative hypothesis and speculated that activation of intimal dendritic cells would induce expression of chemokines and chemokine receptors that are involved in dendritic cell migration, such as CCR7, and that this process may actually protect the artery wall from intracellular pathogens. So this hypothesis was based on the fact that in peripheral tissues, dendritic cells induce the expression of CCR7 and then migrate through afferent lymphatics to regional lymph nodes where they encounter a T-cell repertoire and present antigen to T-cells. Unlike the peripheral tissues, there are actually no lymphatics in the inner wall of the artery, in the arterial intima or the media, and thus we speculated that the cells would have to move through the endothelium in a reverse direction in contrast to what usually occurs in inflammation, where leukocytes usually move through the endothelium into the extravascular tissue. So to summarize our findings, what we actually found was that systemic stimulation with toll-like receptor or nod-like receptor ligands would induce these intimal dendritic cells that reside below the vascular endothelium to express the chemokine CCL19 and as well as its receptor CCR7. And this triggered a migration in the reverse direction through the endothelium back into the arterial blood. When we looked at this process in more detail, we actually found that there were two waves of migration. The first one would occur within 6 to 12 hours after the systemic stimulation, and then there was a recovery in the number of intimal dendritic cells, precisely back to the level that occurred, a number of cells that was found during homeostasis. And then there was a second drop that occurred in the case of LPS, which is TLR4 ligand, between days 2 and 3, followed by another recovery. With poly-IC, which is a TLR3 stimulus, again, there was a drop that occurred in the first 24 hours and a second drop that occurred between 48 and 72 hours. This recovery that occurs after each drop in the number of cells in the intima is due to proliferation of the remaining cells in the intima. And we showed this by pre-labeling the population with BRDU, chasing out the free BRDU, and then analyzing at days 1, 2, and 3, and 4 after a stimulation with LPS. So you can see that at day 1 there is a drop in the BRDU labeled cells. Then there is a recovery. So in other words, these cells had proliferated and the number of BRDU cells increased. Again, there's a drop and again recovery. And with pulse labeling, you could see there's a spike. This is 3-hour pulse labeling prior to sacrifice. You could see that there's a spike in the BRDU labeling index that occurs and coincides with the recovery period. So then we wanted to see, well, what's the purpose of this process of reverse transmigration? And initially we speculated that it may be to remove pathogens from the artery wall. So we injected mice with E. coli, and to our surprise, we found that there were no E. coli in the artery wall to speak of. So in that, we went back to the literature, and we saw that when people using experimental models, when they want to induce bacterial endocarditis, they actually have to damage the arterial endothelium in order for the pathogen to establish an infection in the vasculature. So then we switched gears and we focused on an intracellular pathogen known as Chlamydia muridarum. So chlamydia has been implicated in atherosclerosis and has actually been found in human atherosclerotic plaques. So when we inject in chlamydia, this causes reverse transmigration of the dendritic cells, but simultaneously the chlamydia infects circulating monocytes. And these circulating monocytes are recruited to the artery wall, and then they either differentiate into dendritic cells or they spread the infection further to the resident dendritic cells. And then this second wave of reverse transmigration removes the chlamydia from the artery wall and clears the pathogen from the arterial intima. Thus, that protects the artery wall from an inflammatory response, which probably is not a good thing in the evolutionary sense. So when we then studied reverse transmigration in LDL receptor deficient mice, there were two groups in this. One is a standard chow-fed mice, and these mice do not develop hypercholesterolemia. So here you can see the two drops in intimal dendritic cells coinciding with reverse transmigration, both after injection of LPS or after injection of chlamydia. But when these animals are fed a cholesterol-rich diet for just a period of 1 week, the reverse transmigration is inhibited. And if one studies the extent of pathogen retention in the artery wall by microdissecting the intima and carrying out PCR for the chlamydia 16S ribosomal RNA, you can see that initially it's detected at days 1 and 3. In the control group or the standard chow-fed group, but coinciding with the second wave of reverse transmigration at day 4, there now is clearance of chlamydia in this group here observed at day 5, whereas in the hypercholesterolemic mice, the chlamydia persists. So in summary, we found the homeostatic function of intimal dendritic cells is to protect the artery wall from intracellular pathogens by undergoing CCR7 and CCL19-dependent reverse transendothelial migration. Hypercholesterolemia inhibits reverse transmigration, and these findings may explain why intracellular pathogens have not been identified in normal arteries but have been found in human atherosclerotic lesions. The third section deals with the cellular and molecular mechanisms of atherosclerotic lesion initiation. So the way that I look at atherosclerotic lesion is I subdivide them into different stages. And some of these stages we identified with our own research. So the first stage occurs during the first week of hypercholesterolemia. I call these nascent lesions. And the second stage is early myeloid cell lesion. This is before one sees necrosis of macrophage or formation of a fibrous cap. And these are the two stages that we have been studying to the greatest extent. So nascent lesion formation actually depends on these intimal dendritic cells that normally reside at sites predisposed to atherosclerosis. What happens is within days, these cells accumulate intracellular lipid, here stained with Nile red, and you can see that the dendritic cells stained with CD11C in green, and you could see the lipid accumulating within these cells and even in the dendrites here. And as the lipid accumulates, the cells lose the dendrites, and they become these typically rounded foam cells. So here's an electron micrograph taken at 5 days after initiation of hypercholesterolemia. This is the internal elastic lamina. Here you've got the endothelium stretched up over this foam cell that is bulging into the lumen. So this process occurs quite rapidly. And in order to test the role of these intimal cells that normally reside in the intima, we utilized the model developed by Stefan Jung, where CD11C promoter drives the expression of diphtheria toxin receptor, and thus we can efficiently eliminate these cells from the arterial intima. So here's the kinetics of this process. One day after injection of diphtheria toxin, these cells are virtually gone, and then there's a very slow recovery. By day 7, only 20% of these cells are recovered. By 2 weeks, it's somewhere around 3 weeks, 75% or so. This is in contrast to the spleen, where the dendritic cells recover within 3 days after injection or after depletion by diphtheria toxin. So we bred the CD11C diphtheria toxin receptor transgene into the LDL receptor background. We then fed these mice a cholesterol-rich diet to initiate hypercholesterolemia, and we looked at lipid accumulation. And you could see that in the dendritic cell depleted mice, there was a significant reduction in lipid accumulation, confirming the fact that these myeloid cells are important for the retention of lipid in the arterial intima. And in fact, we didn't find the myeloid cells there. We've found now that the lipid, unlike the previous EM that I showed you where you had lots of these foam cells, now the lipid is bound to extracellular matrix in the arterial intima with the appropriate dimensions consistent with either aggregated LDL or VLDL. So that's the first stage of atherogenesis. Within days of hypercholesterolemia, the resident intimal cells accumulate lipid. And we're still not sure whether this is actually a protective response or a pathogenic response. So the next stage is the early foam cell lesions. As you can see here, these cells are positive for CD68. Now you can see that there's many layers of cells that accumulate. There's still no evidence of necrosis within these lesions. They're positive for CD68, MoMA2, which is another macrophage marker, and as well as CD11C. And we've been interested in what determines what factors or what cellular processes are relevant to the accumulation of these cells in the arterial intima. One can think of the atherosclerotic lesion as this red box here, and there's a potential for monocyte recruitment, cell proliferation, cell survival or cell death, as well as cell egress back into the blood to determine the quantity of or the extent of accumulation of myeloid cells in atherosclerotic lesions. So we developed assays to quantify some of these processes. One of the assays that we developed was BRDU pulse labeling, which not only can be useful in assessing cell proliferation, but it also can be useful for determining monocyte recruitment into early atherosclerotic lesion. So the way that this assay works is you inject an animal with BRDU, which has a very short half-life and is cleared within a matter of minutes. All the labeling cells in the body take up BRDU. So if you look shortly after your injection, you will be able to quantify the number of cells that are in the S phase of the cell cycle. If you look in the blood after injecting BRDU, you will see that there are no circulating cells that are BRDU labeled for several hours after BRDU injection. That's because the cells that are labeled in the bone marrow are immature precursors, and it takes time for them to mature before they are released into the blood. And it was quite fortuitous for us that the first cell that matures and is released into the blood is the classical monocyte, the LY6C high monocyte. And this cell now appears in the blood after about 6 hours after BRDU labeling. And in fact, we could label 40 to 50% of these cells with a single pulse of BRDU. So if one looks in the arterial intima, 2 hours after BRDU injection, one can detect the cells that are locally proliferating. If one looks 24 hours later, one now quantifies the cells that both locally proliferated and that have been transported in monocytes from the bone marrow into the arterial intima. And so we quantified proliferation and recruitment in early atherosclerotic lesions. And you could see that in normal intima, we found that there's generally low level of proliferation, a little bit of monocyte recruitment, but it took 2 weeks of hypercholesterolemia. I showed you data before where after 5 days, there's already lipid accumulation in myeloid cells that reside in the intima. But for proliferation and for recruitment to increase in atherosclerosis, it took 2 weeks of hypercholesterolemia, which is actually, to me, a very astounding finding, that the system works like a capacitor. It takes time. It's not like injecting LPS. Normally, if you have a bacterial infection, let's say in the peritoneal cavity, within 24 hours, you can recruit 10 to the 7 neutrophils. It's a tremendous inflammatory response. Yet in atherosclerosis, it takes 2 weeks for this process to start. And in fact, some of the work that Gwendolyn Randolph has done on reversal of atherosclerosis has showed that it takes 2 weeks for the process to stop. So that's still unknown why it takes so long for the inflammatory response to start. In this study, we actually showed that one of the factors that's responsible for proliferation in early atherosclerotic lesions is GM-CSF, using antibodies to block GM-CSF and by injecting in GM-CSF, showing that this will induce proliferation in atherosclerotic lesions. Back to the point where I talked about the fact that the inflammatory process takes such a long time to develop in atherogenesis. When we looked at gene expression in early atherosclerotic lesions, it took 2 to 3 weeks before we were able to see an increased production of chemokines in atherosclerosis, and there was also an increase in adhesion molecules. So recently, we've carried out a study where we looked at the effect of lipid loading on macrophage gene expression. And if you read the textbooks will say that the thing that drives atherogenesis is lipid accumulation in macrophages. And then these macrophages produce chemokines, growth factors, et cetera, et cetera. But if you take macrophages in cell culture, you load them with lipid. In our case, we used oxidized LDL, which would load them through a scavenger receptor. We found that this had minimal effects on pro-inflammatory gene expression. In fact, what we found was that if you stimulated these macrophages now with a variety of stimuli, such as TLR4 or TLR3 ligands, LPS or poly-IC, in fact, the lipid loading dampened the inflammatory response through some sort of epigenetic mechanism, and we're working on trying to understand what that is. So what's the role of adhesion molecules? Adhesion molecules are important for anchoring the recruited leukocytes to endothelium. A long time ago, I showed that VCAM1 was preferentially expressed in atherosclerotic lesions. We showed that in VCAM1, We developed VCAM1 hypomorphic mice since the knockout was an embryonic lethal, and showed that atherogenesis was reduced in these animals. And recently we used this BRDU assay to prove that VCAM1 and its ligand alpha-4 integrin are important in monocyte recruitment to atherosclerotic lesions, since blockade with an antibody to alpha-4 integrin reduced the extent of BRDU labeling after 24 hours in atherosclerotic lesions. And this is important to do because other studies have developed incorrect conclusions based on their favored molecules. For example, when people who are studying a chemokine known as MCP1 or CCL2, they found that there was a reduction in atherogenesis in these mice. And since chemokines are important for activation of integrins and recruitment, the assumption in the field was that expression of CCL2 at the site of atherosclerosis is important for recruiting monocytes to atherosclerotic lesions. And only 10 years after these high-profile papers were published did people realize that CCL2 and its receptor CCR2 are important for the release of these classical monocytes from the bone marrow. And so this is probably why there was such a dramatic effect on atherosclerotic lesion formation in these knockouts, the release from the bone marrow rather than the local recruitment. When we studied the effect of, when we studied apoptosis in early lesions, we didn't find any increase in apoptosis until 8 weeks after. So in summary, what we feel are the key mechanisms for atherogenesis is in early lesions are monocyte recruitment, cell proliferation, and there is some capacity for these increased number of macrophages to survive since they do not undergo cell death. As I showed you before, hypercholesterolemia seems to block the egress of cells from atherosclerotic lesion. This is based on our work and as well as work of Catherine Moore. So what are some of our future directions? Well, we'd like to discover how the abundance of intimal dendritic cells is regulated in the normal intima. We would like to identify molecules that regulate the crosstalk between endothelial cells and dendritic cells and establish this niche. So we have preliminary data, and we speculate that it's the endothelial cells that are, and the hemodynamic disturbances that drive production of survival factors that allow a certain abundance of these cells to reside in the arterial intima. Although there is recruitment of monocytes, we do not know what the fate of these cells is. We don't know whether these cells contribute to this dendritic cell population or whether they undergo reverse migration or whether they undergo apoptosis. We would like to know what changes in the arterial intima upon hypercholesterolemia and what allows an increased number of myeloid cells to survive and be maintained in this cell. So the people who study macrophages are now, the field is very aware of the fact that embryonically derived macrophages are important in many tissues for providing tissue-resident macrophages. And we need to determine in the normal intima the origin of our cells and whether we're dealing with a uniform population or several population of dendritic cells and better understand how this population is maintained. My colleague Clint Robbins, when he was a postdoc, published that the renewal of macrophages in advanced lesion is dependent not on monocyte recruitment, but actually on local proliferation of macrophages. So something changes between early lesions where monocyte recruitment is important as well as proliferation is important in maintaining the macrophage population, whereas in advanced lesions, this changes to being dominated by proliferation. So we'd like to be able to understand what changes somewhere in our models between 8 and 12 weeks to shift from one type of biology to this type of biology. And finally, we'd like to identify molecular mechanisms which regulate RTM. We know that in addition to CCL19 and CCR7, there are many other molecules that are involved in this process. We'd like to sort out which of these processes are inhibited during atherogenesis, and then to be able to rescue RTM in hypercholesteremic mice in order to actually transport lipid out of the arterial intima and reduce the burden of macrophages in atherosclerotic lesion. So I'll stop in that at this point and acknowledge members, current and former members of my laboratory, as well as a number of collaborators and support from granting agencies over the years. Thank you very much.
A real tour de force lecture on the field. Myron, thank you. Any questions? Myron, maybe I'll begin. You touched on an area that you want to focus on, which is what are the factors that drive the number of dendritic cells. It was quite interesting how, I think the absolute number was 600, I don't know the units, how the recovery after LPS or after chlamydia always sort of reached that plateau. Did that vary from animal to animal, or I'm assuming there are more cells but it didn't look like, even in the hyperlipidemic cholesterol fed, it was always that same cell abundance. It's like it's locked in. Can you comment on that?
Yeah, so we were very intrigued by exactly that observation, and we think that the number of these cells is precisely regulated. Nobody understands what regulates the abundance of macrophages in any tissue. You know, why is there a certain number of Kupffer cells? Why don't they just continue expanding? We suspect that this complex interplay between the local tissue environment and their various cues that are provided by that environment, factors such as MCSF, and different tissues may have different cues. I think we have a pretty simple system where we can quantify the cells relatively readily. There are not that many cell populations there to study, and now with the new genomic approaches, single-cell RNA sequencing, we should be able to get a handle on this, assuming our limitation is that we're working with a very small number of cells. And it makes traditional biochemistry and molecular biology very difficult to do. And so hopefully we need to make some advances to be able to adopt some of the latest genomic technologies, and then I think we will be in good shape to answer these questions.
Any other questions? Jason.
I just had a question about the failure of the cells to migrate when they're loaded with cholesterol. Is that a defect of like the TLR or NLR signaling itself, or is it just transducing that signal into some sort of migratory response?
Yeah, we're not sure why it is. There have been observations by others showing that cholesterol loading inhibits cell migration, period. So it could be that the cells, that this loading affects the cell cytoskeleton or small GTPases that are required to modulate the cell cytoskeleton to promote migration. So it could be a multifactorial process, and it probably is.
Okay, so we'll have a 15-minute coffee break, and then we'll return for our other keynote lecture from Professor Ronnie Cohn on gene therapy.